The United States Center for Disease Control cites antibiotic resistance as one of the greatest public health crises of our time. Each year in the U.S., over 2.8 million people gain bacterial infections, some of which have devastating consequences. In recent years, contracting methicillin-resistant Staphylococcus aureus, or MRSA infections, have been linked to beaches in Florida. Kevin Pettis, a Tallahassee schoolteacher, contracted MRSA after visiting St. Teresa Beach on the Gulf Coast of Florida. This infection resulted in osteomyelitis, a bone infection which caused the amputation of his toe. Pettis claims he contracted MRSA from Florida's waterways. Gary Burris, an environmental filmmaker, developed MRSA skin boils after filming on a Fort Myers beach. He calls on Florida to reevaluate water quality and start testing the water for antibiotic-resistant bacteria like MRSA. This year, our iGEM team has chosen to address this advancing issue affecting Florida's civilians. When we began researching antibiotic resistance, we came across a longitudinal study by Adam Schaefer, an epidemiologist from Florida Atlantic University. This study was conducted in the Indian River Lagoon, a grouping of the Mosquito Lagoon, the Banana River, and the Indian River, covering 156 miles of Florida's east coast. Schaefer looked at bottlenose dolphins, comparing antibiotic resistance from bacteria found from these dolphins over a 13-year period between 2003 and 2015. Their team tested the bacteria for resistance against 15 antibiotics, including ampicillin, ciprofloxacin, erythromycin, and tetracyclines. Overall, in the 2015 sample, over 50% of all isolates were resistant to ampicillin, augmentin, cephalothin, and erythromycin. The multiple antibiotic resistance, or MAR index, is a measurement of antibiotic resistance. It is the number of antibiotics to which an organism is resistant to the total number of antibiotics to which the organism is exposed. As a standard, a MAR index above 0.2 is indicative of anthropogenic or human pollution. In the dolphin study conducted between the two separate time periods, Pseudomonas aeruginosa started at a MAR index of 0.54 and increased to 0.63, while Vibrio alginolyticus started at 0.32 and increased to 0.37. Not only did these bacteria originally have high MAR indexes, but they also increased significantly over the 12-year time period. According to the study, the trends of resistant bacteria on the dolphins seem to mirror the trends of antibiotic resistance in human healthcare. Dolphins are a sentinel species, meaning they can serve as warning to humans for possible future health risks. After finding this study, we wanted to find out how the antibiotic-resistant bacteria were reaching the dolphins. When interviewed by The Guardian about his research, Schaefer claimed that the antibiotics, these drugs themselves, are getting into the aquatic environments, and that's because most sewage treatment doesn't necessarily break down these medications. Antibiotic use in agriculture also likely contributes to the amount of resistance in the water. Usage in livestock and aquaculture allows pharmaceuticals to be released into nearby bodies of water. We chose to focus on wastewater treatment plants. The established conditions and systematic operating of wastewater treatment plants would allow us to develop a solution that worked uniformly across all plants. This is in comparison to independent farming in agriculture, where a solution may not work for every farm. Treatment plants have also historically been placed alongside the Indian River Lagoon to allow for quick and efficient surface water discharge. If the effluent contains harmful contaminants, such as pharmaceuticals, then the treatment plants could be contributing to environmental damage within the lagoon. According to a 2007 report by the Florida Department of Environmental Protection, 475 domestic wastewater facilities in the state treated 1.534 billion gallons of domestic wastewater per day. The acceptable levels of contaminants and treatment processes depend on the fate of the water. The treated water is either disposed of into nearby bodies of water or reclaimed and reused. Florida's reclaimed water is generally used in golf courses, schools, and residential communities for irrigation. The Florida Department of Environmental Protection, or FDEP, is known to test the effluent of local state treatment plants for various pollutants and other constituents. We attempted to find a record of antibiotic levels in the effluent of wastewater treatment plants in Florida. First, we contacted David D. Whiting, the Deputy Division Director of Laboratory and Water Quality Standards Program. He commented that it is unlikely the department is monitoring for non-regulated compounds such as antibiotics. After reaching out to countless other FDEP departments and employees, no one could tell us if a record of antibiotic levels exists or if antibiotics in the wastewater are even being tested for. Not testing for antibiotics or antibiotic-resistant bacteria could have detrimental effects on public health. This brought us to Mike Kelly, an operator of the domestic South Shores wastewater treatment plant in Brevard County, Florida, located on the Indian River Lagoon. Kelly was also concerned about antibiotics in the water.
Unfortunately, we don't know. We do know that there are a lot of pharmaceutical and other molecules that are in the water that this process doesn't remove. And we know for sure that they're in here.
In a meeting, he confirmed that the FDEP does not test wastewater effluent for any pharmaceuticals, including antibiotics. When considering possible solutions to this problem, our team was split between two different options: an algae-based filter or a bacteria additive. The algae solution seemed plausible because algae is known to absorb antibiotics and antibiotic-resistant genes. Implementing the organism with a scrubber or bioreactor and pump could help to filter antibiotics and antibiotic-resistant genes out of the water. The bacteria solution would consist of a genetically engineered bacteria that could produce an enzyme which would break down antibiotics within the treatment plant. Upon further research, we found a previous iGEM project, UCAS, from Beijing, had demonstrated a similar solution which could break down tetracycline using a genetically engineered E. coli that produced the TET-X enzyme. When presented with both options, Mike Kelly strongly preferred the genetically engineered bacteria idea.
Good. We want to make sure that, private treatment plants, municipal treatment plants are all going to get a similar solution that works equally.
Then your best bet is the bacteria.
Okay.
Since microorganisms are already used within the wastewater treatment process as regulators, he thought that the bacteria solution would be more accepted among other treatment plants. Along with this, legal permits, maintenance, and funding could stand in the way of implementing the scrubbers, bioreactors, pumps, and other equipment that come along with the algae solution. Here is an example of a shopping catalog provided by Kelly, where different microorganisms are marketed to wastewater treatment plants. Dr. Yuning Tang, an environmental engineering professor here at Florida State University, also holds a degree in wastewater engineering. When contacted, he agreed with Kelly and opted for the genetically engineered bacteria solution. Tang also confirmed that antibiotics are not being tested for.
To create a genetically engineered bacteria that combats the spread of antibiotic resistance, we have to understand how antibiotic-resistant genes are spread. Conjugation is the most common method of gene transfer within treatment plants. This would make it extremely difficult to attack the antibiotic-resistant genes that exist within each cell. We chose to target the antibiotics themselves, rather than the antibiotic-resistant genes. There is more existing literature and documentation on the impact of antibiotics in wastewater. The specific antibiotic we chose to target was erythromycin. Erythromycin is one of the most commonly taken antibiotics. It is commonly used to treat bronchitis, pneumonia, pertussis, syphilis, chlamydia, urinary tract infections, and skin infections. In Adam Schaefer's dolphin study, they found bacteria resistant to this specific pharmaceutical. To eradicate erythromycin in wastewater treatment plants, we created sewage purification limiting antibiotic spread in habitats, or SPLASH. Our product is a genetically engineered E. coli that produces the enzymes EREA and EREB to target and break down erythromycin. We chose to use erythromycin esterase types 1 and 2 because they remain relatively stable within the pH levels of wastewater treatment plants that we are targeting. These enzymes work by enzymatic hydrolysis of the macrolactone ring. E. coli was selected as our chassis because it is a well-studied and understood organism. The enzyme generator starts with naturally abundant RNA polymerase. Both EREA and EREB DNA sequences are present in the generator. This allows both EREB and EREA enzymes to be produced. Genetically engineering a bacteria with the capability to produce enzymes that degrade antibiotics would give this bacteria an antibiotic-resistant gene. This could pose a threat to the environment if the bacteria were able to escape the treatment plants, and this would worsen our problem. Our bacteria can transfer its altered gene to other bacteria by conjugation or transformation, some of which are pathogenic to humans. To overcome this, we implemented the toxin-antitoxin, or TA module kill switch. This sequence would be attached onto the same device as our EREA and EREB generator. Originally, we wanted to accomplish this using a methane-inducible promoter, as methane is abundant within the components of wastewater. However, we were not able to find a suitable promoter for this purpose. To overcome this, we adapted this methane-inducible system from a 2014 iGEM team, von Schweib.
First, a methane monooxygenase, or SMMO generator, uses a constitutive promoter to produce 6 subunits of monooxygenase. These subunits combine to make an SMMO complex. Methane can be found in wastewater treatment plants. Therefore, methane from outside the cell can react with the SMMO complex to form methanol. A methanol dehydrogenase generator is also employed with a constitutive promoter to produce methane dehydrogenase. Methanol dehydrogenase catalyzes the reaction between methanol and NAD+ to form formaldehyde. NAD+ is already available in the cell, as it is naturally produced by E. coli. NADH and H+ are byproducts of this reaction. Formaldehyde is then used as the direct inducer for the TA module, as a formaldehyde-inducible promoter is used to generate the PAR-D2 antitoxin. The PAR-E2 toxin is produced from RNA polymerase in this generator. When both the PAR-E2 toxin and the PAR-D2 antitoxin are produced, they bind together to form a heterodimer. When the antitoxin binds to the toxin, it inactivates the toxin, rendering the complex completely harmless to the cell. Outside of the treatment plant conditions, where formaldehyde is not available, the PAR-D2 antitoxin will not be produced. Therefore, the PAR-E2 toxin will not be inactivated. The toxin is long-lasting, whereas the antitoxin is degraded relatively quickly. If the bacteria stops expressing the antitoxin plasmid, the toxin will remain inside the cell. This will allow the bacteria to kill itself outside of the treatment plant environment. A downfall of this solution is the presence of formaldehyde in treatment plants. This chemical can cause skin irritation, respiratory problems, and can even be fatal if ingested in small amounts. The primary safety concern of the presence of formaldehyde comes when the water is reclaimed. Along with this, methanol, a highly flammable chemical, will also be produced throughout the process. After meeting with Dr. Tang to discuss these concerns, he claimed that methanol will likely be consumed by other bacteria within the treatment plant. Formaldehyde in the system will persist only at low concentrations. To build our design, we would have built plasmids using IDTG blocks in the New England Biolab's Hi-Fi DNA assembly mix, which provides a one-pot scarless assembly method. We would have designed and synthesized regulatory G blocks that included the BioBrick prefix, a promoter we were testing, a ribosome binding site, and a 25 base pair overlap of the coding sequence. We also would have designed coding sequence G blocks, which included the coding sequence, a terminator, and the BioBrick suffix. We would have used the PSB1K3 plasmid backbone. We expect that over the summer we would have been able to verify the protein expression of the individual genetic devices. We would have tested the 6 listed devices by using E. coli NEB Express as the cellular chassis. The constitutive generators are expected to express the target protein without induction. The formaldehyde-inducible PAR-D2 generator would be induced with formaldehyde, and the expressed proteins would be purified using chromatography. We would have verified the presence of the target protein via microfluidic protein electrophoresis. Splash was designed to be implemented in the bioreactor of a wastewater treatment plant. The majority of bacteria within treatment plants is housed in the bioreactor. However, our solution could be used in many different sectors of treatment. This project was specifically designed with the financial and physical limitations of the wastewater treatment plant in mind. Ideally, the bacteria can be introduced into the bioreactor and then left to degrade the antibiotics. This makes it easy for the plant to implement, since they do not need to make any structural alterations. A flaw of our design is that only macrolides are targeted by the engineered bacteria. However, allowing the bacteria to grade other types of antibiotics would reduce its chance at competing with other bacteria in the wastewater treatment plant. Modeling is an important part of our project, as it analyzes our design in order to determine the ideal amount of bacteria to introduce into the wastewater treatment plant. Our MATLAB modeling design is composed of a series of equations. To model the growth rate of bacteria, we used an exponential equation based on the doubling time of E. coli. The doubling time of E. coli varies from as little as 17 minutes in ideal conditions to as much as 15 hours in the wild. Because we estimate that the water in the tank of a treatment plant will have sufficient nutrients for the bacteria to comfortably reproduce, we set the doubling time to 4 hours. Additionally, the amount of EREB or EREA in the system is proportional to this equation. In collaboration with Team University of Florida, we were able to track down a study from Wenjung Liu that recorded the influx rate of erythromycin into E. coli. This aided in calculating the rate of influx of erythromycin into the cell based on concentration using this equation. In order to properly model the interaction between erythromycin and EREB or EREA, we utilized the Michaelis-Menten equation, which describes the enzymatic reaction rates based on the concentration of the substrate. From this equation, we were able to determine how the rates of reaction changes as the concentration of substrate decreases. With this equation, alongside experimental data from a study, we were able to determine an average rate of reaction between the EREB or EREA and the erythromycin. We also took into account the dilution of the enzyme once placed in the treatment plant tank using a series of equations. Vmax is the theoretical maximum rate of the reaction, and Km is the Michaelis constant. These values are normally determined experimentally. From our model, we were able to determine that as little as 0.8 liters of the solution containing the modified bacteria would need to be added to a 5,000-liter tank in order to eliminate the erythromycin in the given time frame. From these results, we could develop a business plan for implementation. This graph shows the concentration of erythromycin inside of our modified cell. The curve of the graph represents the cells taking in erythromycin and then degrading it with the enzyme. This graph shows the total concentration of erythromycin in the water tank. It decreases significantly over time. For the bronze medal requirements, we added randomized promoters and terminators to the biofab library of parts on the registry. To achieve the silver medal, we collaborated with the University of Florida and ICER Therapody iGEM teams, analyzed the impacts of antibiotic resistance on the environment, consulted experts on the impact of our topic, documented the parts of our solution, and proposed an implementation plan for our project. Finally, to satisfy the gold medal requirements, we created a model to predict how effective our bacteria would be, raised awareness of synthetic biologies to high schools throughout Florida through science communication outreach, and based our project around input from stakeholders. Antibiotic resistance has proven to be a major problem across the globe. Our genetically engineered bacteria will reduce the levels of macrolides in wastewater treatment plants by producing an enzyme to break down these pharmaceuticals without posing a threat to the environment. With Splash, we hope to help not only dolphins but also humans by preventing antibiotic resistance in other habitats. Thanks for watching, and we'll see you at the Giant Jamboree.